Hello, everybody. Welcome. Well, we are right at the hour, and I do like to start on time, so I'm just going to keep an eye on letting people in from the waiting room as people continue to join. And I'd like to welcome everybody to today's Tea Talk. It's lovely to have so many folks joining. I am Natasha Duarte. I'm the director of the Composting Association of Vermont, based here in Heinsberg. It's been a little while since we've had one of these Tea Talk Lunch and Learn webinars, but it's great to kick off a series again. And today we are going to be talking about compost microscopy, matching biology to soil and crop needs. And John Culpepper from Compost for Good is here with us today. This is part of a larger project, which is on-farm community-oriented food scrap composting in Vermont, New Hampshire, and New York. Compost for Good is our New York partner, so it's great to just continue this collaboration. The project is funded by USDA Rural Utilities Services Solid Waste Management Program, and our overall goal for this project is to develop and sustain community-oriented composting sites with farmer partners to manage food scraps, farm residues, manures, and other organics. So we've been working over the last 3.5 years or so to establish and really promote a community of practice, both among our composting partners, but also among the technical service providers who are supporting this work. So folks like those at Compost for Good. And so with that, welcome and acknowledgements. I'm going to stop sharing my screen and invite John to share his while I introduce him. John Culpepper co-founded Compost for Good to help communities around the world upcycle all appropriate organic materials to reduce greenhouse gas emissions, keep resources in local communities, create business opportunities, help solve water quality issues, and lower the cost of food and fiber production. John has worked in education, farming, facilities management, and research, and is a certified microscopist through the Soil Food Web School, and he is currently working on his consultant certification through the same program. And so with that, thank you so much for joining us today, John. We're really excited and looking forward to your presentation.
Awesome. Can you hear me?
Perfectly.
Great. Natasha, thank you for that introduction, and thank you for everyone who is joining. It's a pleasure to speak with you today. So let's get going. All right, Compost for Good. For those of you who don't know us, are essentially three individuals: myself, my daughter Katie, and Jennifer Perry, who is manages our grants. And Jennifer is also a professional composter and has a business in Redford, New York. We have been a partner of a couple of different not-for-profits in the Adirondacks. We're primarily working with the Adirondack North Country Association at this point. And by the way, if Natasha didn't say so, we are in New York, just right over the lake from right, not too far from Lake Champlain. We work with people from all over, businesses, individuals, entrepreneurs. We work throughout the state of New York. I work with individuals throughout the country and also throughout the world. This is our vision statement for those of you who are interested. We are essentially just interested in helping anyone who is interested in community scale composting or anyone interested in upcycling organics. So I want to start here. I want you to drink this image in and imagine that I take a 1 acre section of this forest and I cut down every single thing from the soil up, and I weigh that, all of that biomass. First of all, can that be done? Yes, it is. Typically, you don't cut a large section like that. You cut a representative sample, weigh that. But let's have a look underground. There's a tremendous amount of things beneath our feet. There is a greater amount of biomass underground than above ground. And this includes not just the roots of the trees and the other plants, but all of the life that is underground, the fungal hyphae, the trillions and trillions of bacteria and other microorganisms. Plants produce sugars and other compounds through photosynthesis. We all know that. We may not all understand that this is, for a plant, a very energy-intensive process. In some cases, actually many cases, the sugars and a little bit of protein and a little bit of carbohydrate that plants produce based on the energy from the sun is pumped down into the roots and then out of the roots and into the soil. And so if you understand that these compounds that are produced through photosynthesis is a very energy intensive process, it makes you wonder, why on earth would a plant dump out 50% or more of the compounds produced through photosynthesis into the soil? Well, the reason why is that plants have co-evolved with microorganisms, and those exudates, those sugars, a little bit of protein, a little bit of carbohydrate that are pumped out of plant roots are there to feed microbes in the soil. Those microbes are absolutely vital to the health of the above ground plants. When we do soil tests, we typically look at physical characteristics, chemical characteristics. What we often don't do is look at biological characteristics. Now many soil tests will do a CO2 evolution test, which simply gives you a sense of something metabolizing in that sample of soil or that sample of compost. But it really doesn't do any more than give you a sense of the fact that there is something that's respiring, something that's living in the soil. We've learned about the importance of microbes to the human health, particularly gut health. We're now learning a lot more about the importance of microbes to plant health. But I want to emphasize that there is a tremendous amount that we still don't know. Not only is there more biomass under the ground compared to above ground, but the diversity of species is astronomically more below ground than above ground. That'll give you a sense of the power of microbes. Most of us know that a gram of soil is about a teaspoon of soil, and many of us have a sense that there are lots of things like bacteria in a gram of soil. But there also can be miles of fungal hyphae in a gram of soil. Now, 10 to the 12th is a pretty big number. Put another way, it's 1 trillion billion microbes, bacteria only in a gram of really good soil or compost. 1 gram of soil or compost can also contain tens of thousands of species of bacteria, not individuals, but species. It's incredibly complex. I'll talk just a minute about the benefits of a robust soil microbiome. Once you, if in fact you have eliminated or reduced the number of beneficial microbes in the soil because of fertilizer practices or cultivation practices, or because of other factors that negatively impact the life underground, once we can reestablish that life, we can reduce or eliminate a lot of the inputs that farmers and other land managers apply. We can also increase water holding capacity through increased pore space and increased organic matter. Now, the only way to build structure in soils is through the interaction of biology. Now, if we can build structure, and if we can increase those pore spaces and the amount of organic matter in the soils, we can reduce or eliminate irrigation. We can also increase water percolation rates. In other words, during rain events, we can reduce or eliminate both the nutrient and the sediment runoff that are causing so much problems in our water bodies. All of the above has the potential to save land managers money. Inputs are costs, and inputs are expensive and becoming more and more expensive all the time. And in fact, if you look at trends over time, farmers typically in many places have been applying more and more fertilizer inputs every year for the last 40, 50 years, yet production, the level of production is either stabilized or even gone down. Now, that's a direct result of damaging the microbial life beneath the soil. Also, carbon sequestration can be done primarily through the building of the miles and many, many hundreds of miles of fungal hyphae. That's where most of the long-term carbon is stored in soils. And by the way, soils are the second largest sink of atmospheric carbon on the planet. The first are the world's oceans, but soils represent a tremendous opportunity to sequester more carbon if managed properly. In fact, farms, 1,000-acre farms, 10,000-acre farms have gone from carbon emitters to carbon sinks in as little as a couple of years. And that's happening in many, many places around the world. This can benefit row crops. Increasing microbial diversity in soils can increase pastures, can increase sports fields like golf courses, other sports fields, and really importantly, and this is not emphasized, I don't think, enough, the amount of land tied up in lawns is very significant in this country and others. And if we could shift just a little bit and make those soils a little more, make the soils in lawns, increase the structure of those soils, improve the structure of those soils, we could increase a lot of the benefits that microbes instill on agricultural fields to residential areas. I'll spend just a minute on the soil food web. This is something that many of you have probably seen, but I want to break it down for you just a little bit. What's important here are a couple of things. Diversity is important. In this picture, we see the soil. You can grow in just bacterial soils, but the plants need a lot of inputs in order to produce. You can grow in soils that are primarily fungal, but don't have the other microbes in there, but they won't grow, they won't thrive. So this particular image is broken down in what's called tropic levels. And tropic levels are essentially energy levels or feeding regimens. And so you can see all the way to the left, bacteria, fungi feed on organic matter and minerals. Believe it or not, fungi and bacteria can solubilize plant nutrients contained in sands, silts, and clays. And that's great to build up bacteria and fungi, but without the other organisms in higher trophic levels, then you don't get the nutrient cycling that you would otherwise. So if you have a lot of bacteria, a lot of fungi in your soils, you tie up a lot of the plant available nutrients in those organisms. The best way to release those plant available nutrients in the bacteria and the fungi is through the predation of higher trophic level organisms on the bacteria and the fungi. So the soil food web, very complex, but simplified, at least for me. This is what it. This is a way to simplify it. Bacteria break down organic matter and minerals. Fungi do the same. Both produce glues, mostly the bacteria, but some glues are produced by the fungi as well. And those glues bind soil particles together. Protozoa, nematodes, and microarthropods come along and they consume those lower trophic levels organisms and release all those nutrients into the soil. So who cares? What does it matter? A lot of studies have been done all over the world whereby soil scientists have gone in and analyzed the microbiology under different plant regimens. And if you survey an area where there's been a lot of soil disturbance or where you have a lot of annual weeds, what you find is primarily bacteria. Moving up that successional plant successional scale, all the way to the far end are old growth forests. And if you analyze the soil beneath old growth forest, what you find is primarily fungally dominant soils. Now, in order to thrive, they still have to have the bacteria, the protozoa, the nematodes, and other beneficial aerobic microorganisms. But this can be really helpful if, in fact, you are growing, for example, perennial plants, blueberries, cherry orchard, whatever it is, grapes, whatever. If you can shift the ratio of fungi in the soil so that you have more fungi than bacteria, then those plants will thrive where they won't thrive necessarily otherwise. On cropping systems, if you can introduce more fungi into the system, then what you tend to have is weeds that don't thrive. Now, this is a little bit of a new concept for many people, the idea that you can combat weeds in cropping systems through changing the complexion between fungi and bacteria is pretty novel, but it works. The stars in this process, all these good things are the bacteria, fungi, protozoa, nematodes, and microarthropods. So let's have a quick look at these. So these images are all under 400 magnification, and I think you can probably see my cursor, but the little bitty dots here, about 1 micron across, are mostly bacteria. So in this one field of view, what we see is some mineral bits, but mostly bacteria. I see maybe one fungal spore in there. If we look at fungi, or if we look at bacteria in motion, you can see them moving around just a little bit, again, at about 400x or at 400x. Fungi and fungal spores are shown in this slide. And if you can see my cursor, this is a fungal spore. This is a fungi, fungal hyphae germinating from that spore. Here's another, if you can see my cursor, another piece of fungal hyphae here. Protozoa. There are lots of different kinds of protozoa. In fact, lots of different kinds of fungi, microarthropods, protozoa. This is one that that is called a testate amoeba. In other words, there's an amoeba inside of a test, and the amoeba actually produces this test and protects that amoeba from predation. So in a healthy, when you're looking at healthy soils or healthy compost, you'll see lots and lots of these. And what you really want to see is many in every single field of view. Nematodes, beneficial nematodes, mostly many of us have grown up thinking that nematodes are bad because there are root-feeding nematodes that do a lot of damage on crops, on trees, on shrubs. But we're talking about beneficial nematodes. They can consume up to 10,000 bacteria in a single day. Many of the protozoa can also consume that many. Let's just take a quick look at a nematode in action here. Again, 400x. The little bitty dots are about 1 micron across. These are bacteria. And here is a nematode looking for bacteria to eat along a piece of organic matter. Microarthropods are exciting when you come across them because they jump out in the microscope and they look like gigantic things. You can barely see them with the naked eye. In fact, many times you can't see them with the naked eye. But they are important, again, for cycling the plant nutrients that are tied up in the lower trophic level organisms, the bacteria, the fungi, the protozoa, and the nematodes. I want to spend a minute talking about soil aggregation because it's incredibly important. Bacteria and fungi, as I've mentioned, produce organic compounds that glue things together. These glues bind organic matter. Uh-oh. That.
Can you get back?
Let me get back to where I started before everyone sees. Let's see here.
There it is. Okay. Perfect.
In the right conditions, these the bacteria produce glues that form what we call microaggregates. Fungi come along, they wrap those microaggregates into larger aggregates that we call macroaggregates. And it is these macroaggregates that produce the structure in soil that allow for deeper rooting, that allow for more water retention, that allow for greater percolation during storm events. So here's an image, again, at about 400 magnification of a micro aggregate just beginning to form. I see most of what I analyze under my microscope, and I analyze lots of samples from farms and other land managers, most of what I see is bacterially dominated soils, bacterially dominant soils. And most of what I see are micro aggregates if I see that. What I really want to see is this. This is the formation of macro aggregates. Here's a big piece of macro aggregate. You could see a fungi growing out of that. If I were able to slice this organic clump apart, I would see lots of other fungi wrapping the micro aggregates together. So nutrient cycling, how does this work? And by the way, this is hard. This was really hard for me to believe when I first heard it. There's enough plant nutrients, phosphorus, for example. There's enough phosphorus in soils all over the world, just about any soil that you want to look at, to effectively grow crops for 1,000 years. So why do we feel like we continually have to add inputs like phosphorus, like nitrogen, like potassium, and like other plant nutrients? Well, it's because those nutrients aren't available to plants, and so we've created these conditions whereby soils don't have all this life, and therefore aren't able. The plant roots, the plant root hairs, aren't able to uptake what they need when they need it. And by the way, what a plant needs changes on a minute to minute, probably second by second basis. And many of those compounds, those exudates that are coming out of the plant roots, are signals to specific microorganisms to, I don't want to be too anthropomorphic here, but to go out and find me some molybdenum or phosphorus or nitrogen or whatever, and the microbes will follow that signal back to the root hair and will provide whatever that plant is looking for. So the biology in the soil has a tremendous amount of plant nutrients stored up in them. And going back to that slide where a gram of soil has, this enormous amount of bacteria, each of those bacterium have plant available nutrients in them. So how do you get those? How do plants get to those nutrients? Well, those bacteria have to be consumed by other organisms in order to put those nutrients into the soil water, so that plants can get to them. Plants are then able to uptake these foods into their roots. So how do we establish biology in our soils, and if we need to, and then how do we keep that biology in the soils? Well, the easiest, the quickest, the best way, I don't know about the best way, but the quickest way to do it is through the development of highly diverse compost, diverse in terms of the microbiology in the compost. So for the purpose of this talk, think about, and the way I think about compost when I'm making compost is I'm actually farming microbes. That's what I'm doing. I'm growing microbes, and I'm doing that for the benefit of the plants growing out of the soils that I put those microbes on. So there are a lot of different ways to break down organic matter, but what we're going to focus on is aerobic decomposition. Anaerobic decomposition, that which happens in the absence of oxygen, is not what you want. You want decomposition that happens in the presence of oxygen. So You want to create as many beneficial aerobic microbes as you can. And diversity is key. And how do you determine this? Well, the only way at the moment, or the best way, the least expensive way to do it, is to confirm that biology under a microscope. Now, that doesn't mean that you need to run out and buy a microscope. There are a lot of trained microscopists out there, and if you're curious about how to contact one of these soil microscopists to give you a sense of an assessment of the microbiology in your compost or your soil, then there are ways to do that. I'm happy to talk about that during Q&A. Okay, so not all compost is the same. I've analyzed many, many hundreds of different composts, and I can tell you that there are no two alike. I have a colleague down in the Albany area that sells 1 cubic foot of compost for about $20. Catalyst Bioamendments in California, they sell a cubic foot of compost for $200, a full order of magnitude higher. Who on earth would pay $200 for a cubic foot? Well, these folks are making more compost than they can sell, and they're selling it to farmers. As you may know, farmers don't typically spend money on unproven ideas. The difference is biology. So in order to keep biology in the soil, you've got to have continuous living roots, whether that's cover crops, the crop itself, pastures, whatever. The more part of the year that you can have continuous living roots in the soil, the better. You want to reduce soil disturbance. This is sort of permaculture 101 or regenerative farming 101. And when you reduce soil disturbance, you maintain particularly the fungal communities intact in your soil. And you want to maintain microbial diversity by, to the extent that you can, by feeding those microbes either through living roots or in specialized situations by organic matter slash or compost or whatever that's added directly to the soil. And how do you determine whether you've got good biology in your soil? Again, you've got to test it with a microscope. Okay, just a moment on climate change. And I think this is a repeat of what I said earlier. And I just want to get to the primary takeaway so we can get to Q&A. What I'm offering you is to think about nature-based solutions, not necessarily human-made solutions. Compost is the fastest and perhaps the best way to add biology back to your soil. Think of composting as resource recovery rather than just waste management. In other words, make the best, highest quality compost that you can for sale or for use on your own property. And then maintain continuous living roots if you can, and minimize soil disturbance and feed those microbes. So that's all I've got for you today, and I would love to open it up, Natasha, for Q&A. Hopefully I've left enough time.
Yeah, I think we have about 25 minutes, which is great. And we can open it up. There's just over 30 of us here, so maybe raise your hand if you want to unmute, and then we can do it that way, just so we don't talk over each other. But you're welcome to also unmute if to get us going. And even though I muted some of you, I think you can still unmute yourselves. But we do have a question in chat, and I'll just start with these. But if we need more detail or if you want to add something, just let me know. So Myra says your audio. Oh, we need a fun name for biology. I don't know. Maybe we can circle back to that one, Myra, and you can unmute. And make some suggestions. Karen asks if you could share, John, The last slide or put the contact info in chat. Karen, just so you know, that will be part of the recording, but also we'll post it on all of the contact information for Compost for Good as with the recording and also on the CAV website. And John, feel free also if you want to just clip that and put it in your contact information in chat, or I can do that in just a minute. Melissa says when we talk about biology needing continuous living roots, what are the best recommendations for winter time when the plants have died back? Are the roots, if left in the ground, still viable as living roots that you're speaking about?
That's a great question. The dead roots are going to turn into, in the right conditions, going to turn into stabilized organic matter. So dead roots are great. There are a lot of living roots that even in this latitude we can have in cropping systems, whether it's winter hardy wheat or cowpeas or whatever. Generally speaking, there are and matching a cover crop to a cropping system can be a real challenge, as many of you no doubt know. There's a lot of experimentation going on right now about what sort of cover crops can be used in different kinds of cropping systems that reduce the overall work of the farmer and benefit the plants. So the roots are producing the exudates that are feeding the microbes. Now, if microbes go for microbes can certainly last through months without food. They do that by going into, for protozoa, for example, they go into encysted states. And so those cysts can live in the soil for years, if not hundreds, if not thousands of years before they see the right sort of environmental conditions to then germinate or sprout or get back to a more active microbe. Hopefully that answered the question.
Great, thank you. Let's see. Kat asks if you could talk a little bit about the different types of nematodes and how recent research is showing that compost maturity can be determined by the types and numbers present.
It's a great question. So my current training is through the Soil Food Web School, and what they encourage consultants and microscopists to do, and my certification, my microscopy certification is through the school, is we look at it in terms of functional groups. So nematodes, there are essentially 4 or 5 functional groups. And the only functional group that we would consider, generally speaking, not a great nematode to have in the soil are the root-feeding nematodes. And you tell them by their, primarily by their mouth parts. And it requires magnification up to 400x to really good shadowing to see the mouth parts in order to understand whether they are a bacterial feeder, a fungal feeder, a predatory nematode, or a root feeding nematode. Root feeding nematodes are pretty easy to pick out because of the specialized mouth parts. They have a spear with a pump beneath that spear that you can see quite easily. I would say that compost maturity is when you have good mature compost is when you have a lot of different species of bacteria, fungi, protozoa, nematodes, and microarthropods. So using just nematodes as a measure for maturity, no doubt you can do that. But for me, it's better that I have an understanding of of those other microbes as well, because that's what we really want. Maximum diversity of aerobic beneficial microbes in the soil.
Great, thank you. And Jay, I see your hands up. I'm going to pull one more question from chat and then turn it to you. Um, uh, Dan says lawns aren't going away. They have living roots. So what can we do to increase their value to the ecosystem?
Fantastic question. So if you can, a lot of people spend a lot of money on lawns, right? On inputs, on pesticides, fungicides, herbicides, fertilizer, irrigation. If you can put a little of that money into soil tests, particularly biological tests, if in fact you are missing some of these major groups in your soil, then the first thing you want to do is get that biology back in the soil. And the best way to do that is to find good, biologically robust, or what the Soil Food Web School calls biocomplete compost. And that's compost that's got a large number and variety of bacteria, a large number and a variety of fungi. These are all aerobic protozoa, nematodes, and microarthropods. So if in fact you've been fertilizing and putting things like pesticides on your soils, on your lawn, you could very well be damaging or reducing the species diversity of the microbes in your lawn. So, a radical idea: stop all those inputs and find the best compost that you can, and assure yourself that you've got good compost by having someone who is able to analyze that compost under the microscope.
Thanks, John. And I'll just quickly add for anyone who's interested that on the Composting Association of Vermont's website, we do have some information about how to top dress lawns and turf with compost as well. Not so much on the website, but I can look some up and share it with the recording. There is also some interesting research that shows more diversity in type of lawn. So not just a single species, but a diverse species and mixing in some native species will also encourage that diversity below ground as well and help support that. So those are just some ideas to share as well in terms of maybe trying to get the most out of lawn and while maintaining lawn. Jay, go ahead if you want to. Oops, sorry, John, did you
Could I
want
just
to?
add to that?
Yeah, please.
One of the reasons why I'm so excited to be in this particular space, this field, is because of the opportunities that are out there. And those opportunities are all over the place. I'll predict that within the next 10, 15, 20 years, you'll see, instead of ChemLawn, or I'm sorry, I don't mean to pick on ChemLawn, but instead of seeing trucks that are coming around to spray or fertilize your lawns, you'll see specialized compost extract applicators that will come by and, for less money perhaps, reinvigorate your lawn. And by the way, you don't have to do this every year like you do with standard inputs. Get that biology back in your lawns, and as Natasha said, if you can, mix in some species diversity in terms of grass species diversity. So that's my prediction. A lot of potential opportunities.
Fingers crossed, John. That would be great.
Yes.
Jay, go ahead if you want to unmute.
Yep. Can you hear me?
Sure can.
Yes.
Okay. Yeah, I just wanted to, if it's okay, I just wanted to just throw a couple things out there as an addendum. Is that all right?
Sure.
So I wanted to add that there's some interesting stuff around, when we talk about nutrient cycling in the soil food web model. There are other metabolic pathways in soil systems, biological pathways that bring nutrients to plants. For instance, mycorrhizal fungi that move, phosphorus and nitrogen sometimes as amino acids directly to plant roots they're partnered with, but also endophytic bacteria in the rhizophagy cycle. And so I wanted to throw that out there because, I had recently seen under a microscope, I'd taken a sample of some soil from a coffee plant that someone was growing in a coffee shop, just as a curio, right? And looking at that soil, which was pretty desiccated at the time, you can imagine people are not watering it consistently, right? So it's probably going through some pretty serious wet dry cycles in this pot. And what was interesting to see was what I was witnessing were a lot of fungi that were sporulating, reproducing very heavily, possibly because they were stressed. And then a lot of bedelloid rotifers, which I'd never seen before. They don't live in water. They live on hygroscopic spaces on the outside of leaf litter, for instance, and they were feeding on the fungal spores. So there you're seeing this really interesting form of nutrient cycling that's highly novel, and very specific to this plant in this setting. And so I guess what I'm getting at is there's a lot of work that Dr. Christine Jones is doing on seed microbiomes and the contribution that a seed will actually make to the microbiology that ends up surrounding and working with the rhizosphere of plants. So I think that a couple of things that I've found in the work that I do is that one, if there's one simple important thing that should happen, it's making sure we have aerobic conditions in soil. So whatever type of microbiology, whether it's brought in through compost extract, or the seeds itself, has an opportunity to thrive. And then two, looking at nutrient cycling in soils that's biologically mediated, not just by the crops or the plant community growing there, but for instance, by the soil order itself, like the USDA's taxonomic classification. There's just very, very, very different plant communities and very different soil, physical and chemical conditions that are going to drive that biology. But I guess what I'm saying is that even in a weird, novel situation, what I see is the biology tends to find its way towards some sort of homeostasis that will, if it can, support the plants growing there. So that's something I wanted to throw out, is that there's also these other metabolic pathways and very novel nutrient acquisition pathways in certain situations.
That's a great addition, Jay. Thanks for uh, talking about this. As I mentioned earlier, what we don't understand, there's so much for us to learn, whether you're a hobbyist or a soil scientist. We just really be, scientists like myself always think that, we've got all the information we need to make the right decisions, and then 10 years down the line we think, oh, how could we have said that back 10 years ago? So there's a lot of work to be done, lots and lots of work to be done, and so appreciate that, Jay.
Yeah, thank you so much. That's super interesting. We are going to go to another question from Karen in chat, who asks, is it beneficial to add various fungal inoculants to the soil prior to planting late succession tree species in riparian areas?
That's a great question. What I tend to advise people not to do is purchase specialized inoculums for a couple of reasons. The plants that you're growing, the plants that thrive in the area where you are, they thrive under the conditions of very specific and microbes that live in that area. So, for example, it's usually never wise to take an inoculum made from wherever, California, Australia, whatever, and add that to your soil. A, it may not be very effective, and B, it could be you're wasting money. Now that there's a lot of research to be done there to prove or disprove this point, but your easiest, best, most effective way is to create compost from a wide variety of feedstocks, because each one of those feedstocks, whether it's crop residue, food scraps, manures, each of those feedstocks are inhabited by thousands and thousands of microbes that are specific to the area. And so the best approach, I think, is to work with the microbes that you have, They want to reproduce, given the right environmental conditions, they reproduce like crazy. So you can get all the inoculum that you'd ever want pretty cheaply, pretty inexpensively, and pretty easily.
Yeah, and just
I hope that helps.
a quick addition to that. Well, not so recent, but like maybe in the last 6 months, I've heard a handful of people who actually bought some of these inoculants and then started looking at them before using them. And they bought them online from Amazon or from somewhere else. And it wasn't at all what they were claiming that it was. And so there's a little bit of buyer beware. I'm not saying that there aren't high quality options that are there. But if you're buying things, especially online or from distributors that you don't know, just do your own due diligence as well. But I generally agree with everything John said as well. Kim asks if you have recommendations for best places to send samples for biological testing. I know you do some of that, John.
I do.
Yeah.
I do. And I do most of my work for at no cost to the person sending the samples in because we work Compost for Good works are paid through the grants that we write. But in the grant world, as many of you may know, it's a little dry spell at the moment. So if you go to the Soil Food Web School, there is a map of soil microscopists all over the world, and you can quickly drill down in your particular area to find those who can do microbiological assessments of both compost and soil.
Yeah, and I'm just going to take this opportunity to say I will add a link to that when we post the recording, but also Kat has shared a resource in chat. Jay, you might have resources, really an open invitation to anyone. If you would like to share some of what you're reading or looking at with this community, please feel free to send it. You all should have my contact information. And I'm happy to sort of collate and share some of that out with this group as well. We have about 6 minutes left. I think we can get through a couple more questions. Karen asks if adding bokashi inoculated with EM1 into soil effectively brings the bacteria, fungi, protozoa, and actinomycetes into soils. I don't know if you...
Bokashi is just like any other decomposition process. There's highly biologically robust bokashi compost and some that are not as robust. So I think it depends. I think it depends on what plants you're working with, whether they really thrive in what sort of bacteria to fungi ratio they thrive best in. And this is an area of research. There's a lot of research out there, a lot of experimentation out there, I should say, where folks are using ways to capture the microbiology that are in the soils in their area, especially under or in forested areas. Now, I don't recommend going out and digging soils for inoculum, but you can place containers of material that microbes will migrate into, and you can help yourself that way. But for me, aerobic decomposition, regular old composting is the simplest, easiest way. Nothing wrong with bokashi, nothing wrong with worm composting. In fact, fantastic. All of that is good stuff. So I say use whatever you got available to you.
Right, thank you. Bruce asks, in your experience, John, are there any beneficial microbes that prey on invasive jumping worms or eggs, or any ongoing research efforts that can give hope to those of us who have been invaded?
Yeah, that's a great question. And I don't have a lot of expertise in jumping worms. I do know that they're fairly easy to kill in with temperature and time. So if you've got an area that's infested, and if it's living in the duff, the organic material, which they typically do, if you can round that up and compost that, you can kill those jumping worms pretty effectively. Now you're not going to get them all, and this is one of those invasives that we're going to be battling and thinking about and trying to understand for a very long time. But you can eliminate them, large numbers of them, fairly effectively.
Yeah, and I'll just say, Bruce, that there are a lot of jumping worm resources on the CAV website, and UVM is really leading the way in a lot of research around that. But I think in some cases we're learning to manage the populations and coexist rather than eradicate. Just as John said, there are definitely a lot of a lot of challenges with them. Let's see, just scrolling through. We just have a couple minutes left. I want to just say, because people started dropping off a little bit, I just included a link to the very short evaluation form in chat. Again, it really helps us with funding and with our grant reporting. If you could just take a minute or two to fill that out. You will be getting it by email as well. Let's see. Kat says it's very affirming to hear you indicate that growing your own locally adapted microbes via aerobic composting is the way to go. So agreed there. I did see someone ask about how to make good aerobic compost. That is a topic for a whole different webinar. We also have a number of recorded webinars and resources talking about best practices and compost recipes and systems and management, all of that, all of those different aspects go into making a good, biologically diverse compost. And I think we're going to end there.
Natasha, may I just offer that if anyone
Yep.
has questions and you don't have a time and didn't get a chance to ask them, feel free to email me. I spend a lot of my time responding to folks, and I love doing it. So please do.
Yeah, and so we'll be sure. Thank you for that, John. And likewise, if you can't find certain resources that I've referenced, please, please let me know as well, and we can point you to those. And I'll be sharing John's contact information. I just want to thank you, John, for joining us today and for sharing all of your knowledge around this. Thank you so much again to everybody for joining, for your questions and participation, adding your thoughts.
Thank you, everyone, very much.